ahead, Miranda. Thank you for joining us today.
Yeah, sorry, I can't be there in person. I'll try to jump in and out of the different sessions from now on. So I think that it's really a good follow-up to Charlotte Norris's talk, because the things that she identified, I think aggregate stability, mineralization, and soil organic carbon are all things that are mediated and dictated by soil biology. Similarly, the last question is about what co-benefits are people interested in. I think in terms of the soil, almost all of them are really impacted by soil biology. So I'm going to talk to you about soil biology. I'm a microbial ecologist, soil microbial ecologist. And one of the most common things I find myself talking about is soil health. And that's tricky, right? For me, soil health is just soil biodiversity. That's the easiest answer I can come up with, but it's not very satisfying. And it's hard to actually make recommendations to growers based on this kind of vague concept of soil biodiversity. So I use genomic tools to understand what soil biodiversity looks like. And I guess today, the point of this talk is to show you what some options are, talk about the pluses and minuses of all the different options, and then hopefully decide somehow which is the best set of tools for this living lab project. I don't think it's black and white. I have a personal favorite, but see if you can tell which one it is. Okay, so first of all, we're talking, and by the way, if anyone has questions, please feel free to interrupt me at any point. I'm happy to answer your questions, but I can't see I can't see the comment box, so just speak up. I guess the first place to start is, what do we measure? If we're interested in soil biodiversity, and I know from the community, farmers are very, very, very interested and very committed to soil biodiversity, protecting it. Well, what does that mean? What do we measure? Because if you look at the soil food web, it includes everything that's in the soil and basically everything that's in a terrestrial system and an aquatic system too. So there's like fungi and bacteria, which I tend to focus most on, and which most soil ecologists focus most on. Well, sorry, Tom, there's nematodes down here too. There are other microbiologists who study eukaryotes, protists, nematodes, and macroinvertebrates, and even vertebrates, right? Like there are large animals that live in the soil. So, oops. Oh my goodness, I'm giving away all my, there's my talk, guys. I don't know. Yeah, okay, let's go back to this one. Okay. Sorry, the screen is not cooperating. Anyways, I will just leave it at this because I can't access the tools. Number one, what do we measure? Do we stick with fungi and bacteria? That's my jam. That's what I study, but I don't want to insinuate that the other groups aren't as important. So that's what the first consideration. But then how do we measure? Who's there, but what are they doing, right? So there's two very, very, very important questions, and not all tools answer all of those questions, which I think are fundamentally important. Actually, I need to have my slideshow going or else my animations won't work. How do I get there? Okay. First thing, who's there? How can we tell who's there? Well, we can sequence the groups of interest. Like I said, I do bacteria and fungi mostly. You could do anything you wanted. But for the sake of this discussion, let's talk about bacteria and fungi, because I think that they are very, very key players in all of the main soil ecosystem processes. One of the things you can get from this information, if you just sequence the soil, is you can find out how many taxa are there. So these ASVs, just pretend those are species for this conversation. And I just pulled this from a study that was looking at species richness of bacteria as you go down in the soil depth profile. And obviously you have more species up higher in the soil profile, very few species down at 180 centimeters. Okay, so that's one level of soil biodiversity. How many species are there? That's really, really important because we know from the literature, more species equals more stability equals more functionality. But if you incorporate composition of who's there, not just the number of species, but also who those species are, you get even more information. So this is a graph that shows you each dot or triangle represents one soil sample. And this graph just shows you how different the soil samples are in terms of composition, what bacteria are in these soils. And you can see very clearly the blue ones are at the surface are very different than the ones over here, these kind of turquoise ones that are 3 meters down or 2 meters down. This just gives us an idea of how different they are. Doesn't really give us that much more information. So if you go even deeper into the sequencing, you can find out, okay, if these are your soil depths, let's look at the bar graph first. If you look at the soil depths, surface to 180 centimeters, you can look to see which groups of organisms, which species, which families, which class, whatever, represents most of the biodiversity. So you guys have probably all seen these, right? And it's good. It tells you, relatively speaking, how many of each different taxa you have. You can drill down in that and look at a specific group, right? You can look at if we look at all of bacteria over the soil profile, you can see that they are, you can look at the trends of each specific group, and you can dial down as much as you want into specific species even, which we haven't done here. But you can see some decrease over soil depth and some, like the proteobacteria, increase over soil depth. So you can get some really interesting trends there. So the good thing about sequencing your soil community is you get good resolution. You can get down to the species level, not below that. Gives a good indication of abundance. It's not perfect, but it gives you a really good relative idea of how many taxa or how many individuals in each taxa are there. And you can see how each species changes with treatment. So if we used it for this experiment, the cover crop, the new BMP versus traditional BMPs, you could see how compositionally the soil's changing across those two treatments. So that's all good stuff. The cons, as is true for everything about soil biology, many of the taxa will remain undescribed. So you'll just get unknown species changing, which is interesting, but kind of unsatisfying. Doesn't reflect functioning, right? Just because you know who's there, you don't know if they're active. You don't know how active they are. They could be dormant. They could be dead. We don't know. We just know that they are there or they have been there. Another con is that it's limited to specific groups, right? Your groups are fairly large, bacteria or fungi, but also it's not looking at the whole thing at the same time. And the last con is spatial heterogeneity and hyperdiversity. Now, this is true for all of them, though. You have to sample intensively to get a good picture of what's going on, because the soil ecosystem is so variable in terms of microhabitats and community composition. So you have to sample intensively, which is expensive. And I think we can just have a caveat: all of these tools are very expensive. So that's all I have to say about that. Okay, so there's another way of measuring who's there besides community sequencing, and you can do eDNA profiling, and that's where you sequence everything in the soil. Here, and when I say everything, I mean everything. So you're not just doing bacteria or fungi, you're doing, in this case, it's annelids, arthropods, chordates, cnidarians, gastropods, nematodes, Rotifera, everything. You can tell, everything that has passed through the soil, passed over the soil, been in the soil, or been in the water that's in the soil, you know if it's there. And this is interesting, but I don't know that it's that useful for what we're talking about. Not all groups are represented, may overrepresent large things or dead things. And honestly, for this topic, if we're talking about co-benefits of interest, I don't know. I mean, maybe the farmers are interested in how many moles are on their property or owls. Who knows? But it doesn't really get at soil nutrition or soil stability. So eDNA is interesting, but I don't know if it's right for this project. Okay, so those are the two ways of figuring out using genomics to figure out who's there. The next methods I'm going to talk about are figuring out what they're doing in the soil. And you don't have to know who's there. You could just look at what's being done in the soil. And the easiest way to do this is with quantitative PCR using specific gene assays. So we pick a gene of interest in a pathway of interest, let's say nitrogen cycle. We could target any or all of the genes in the nitrogen cycle to find out how the best management practices affect the cycling of nitrogen in the soil. It's easy, it's really relatively cheap, it's fast. Doesn't tell you anything about the larger community though. Maybe we're not interested in that. The pros is it's cheap, gives high resolution information about specific genes. You really know how much they're being expressed, but it can only tell us about the genes we're looking for, which I feel like it's a pretty big limitation, because I feel like we're at the frontier of understanding, just a very tippy-toe frontier of understanding soil biology and soil ecosystem functioning. And while we understand a few pathways very well, there are many, many that we don't, and they might be really, really important. This won't help us with those pathways. So the second one is the second way of figuring out what the soil organisms are doing is through metatranscriptomics. And metatranscriptomics is transcriptome profiling. So looking at all the transcripts in the soil, but not from one organism, from every organism. So that's why it's called meta. You've probably seen, I don't know if you've seen graphs like this, heat maps. I see them all the time. I find them really overwhelming to look at. So I'm just going to tell you how I look at them. This is a graph that shows transcripts expression in different from soil microbes, and all it means is here on the y-axis we have the organisms themselves, right? The different microbes, whatever they are, it doesn't matter. And then on the top and the bottom we have the gene transcripts. So it shows us that in this group of organisms down here, in this group of genes, transcripts are really overexpressed. These organisms, these pathways were really, really active in certain treatments. Whereas over here in the blue section, they were under expressing. So gene activity was reduced. And the great thing about this method is that you have no preconceived ideas going into it. The data reveals how the soil function changes across treatments, right? And it might be something you predicted, like if you're growing legumes, you might predict changes to the nitrogen cycle, or it might be something completely different that you didn't anticipate. So this is a really powerful method. It gives us a lot of data. And it also gives us really specific data too, right? And similar to qPCR, we can identify specific specific steps in the pathway and understand are they ramping up or ramping down, depending. So if nitrogen limitation is a problem, you can see, ooh, maybe nitrogen turnover is limited at this specific step because something's happening to this particular guild of microbes, or they don't have the substrates they need to perform that reaction. So you can get really high level, highly specific information about pathways. At the same time, you get loads and loads of information. So it's kind of the best of both worlds. So pros, comprehensive information about functional capacity of the soil. You can identify specific steps that are under functioning or over functioning in a nutrient pathway. Cons, it's very, very expensive, which means you can't sample as intensively as you can for the other methods, which if you go back to my earlier comment is you have to sample intensively to know what these systems are doing, across the entire plot of land. There's a lot of information you get from this method, maybe more than we need, but I don't know. I don't know how much we need right now. I think it's important not to anticipate what we think we need to know about the soil. So let's see. So what is the most relevant for this particular study? I don't know. I feel like most of my work so far has been doing soil inventories, finding out who's there. And you can tell a lot about the soil from looking at who's there, but it's unsatisfying because you don't really know what's happening. You can infer based on who's there, how the functioning might change. But I think the farmers, at the end of the day, all they care about is how functional are their soils. So to do that, I think we have to look at either qPCR or metatranscriptomics. Which brings us to my very last question. What can we afford? Can we afford to do metatranscriptomics? I think we can. I think we can on a reduced set of plots. And I think it would be really, really valuable. But that's my presentation. I hope you got the gist of what the field looks like right now and how we might be able to use it for this project.
Taking a pause here and discussing these two questions that Miranda put up, like around what's most relevant, I think it would be really interesting to hear from the people who are involved in the projects, what your thoughts are around the soil health piece. Some of you may already be integrating some of this into your project, for some people it might be newer. Also hearing from ASB what you guys are thinking, and then we can go to Tom afterwards. But yeah, or if there's like specific clarifying questions for Miranda, that's also an option. So for the virtual folks, you can also unmute and ask a question as well. Yeah.
Thanks for that great overview, Miranda. As a microbial ecologist who hasn't taken the leap into transcriptomics yet, and then building on with what Charlotte's saying and the discussion with, it's better to have all this run through one lab. Is transcriptomics, is that feasible to run it through one lab, or would we be processing it in different labs throughout, our own research institution?
It's a really, really good question. And I think even more than what Charlotte was saying, this is so important to have a common pipeline, because there are a million decisions along the pipeline on how you handle your bioinformatics that can dramatically change what your data looks like. So I would suggest that we have a coordinated effort. We use the same sequencing facility, but more important than the same sequencing facility, that's less important. I think the most important thing is that we have a shared pipeline and we use the same bioinformatic tools. But you're right, if we don't do that, then I don't think the data would be comparable at all.
Great, thank you. That's kind of what I was thinking. And hopefully I can piggyback with you.
Do they know who's talking in the room? If they can.
No, that's a good point. So that was
Oh,
Michael
sorry.
Preston
Michael
from
Preston.
University
Sorry.
of Northern British Columbia, who's working on the cattle board project.
Oh, I just emailed you, Michael, this morning. I just emailed you this morning. Hi.
Yeah, and so for the cattle board, if you guys are this plan as part of your projects and you have Mike Preston on board, which is awesome and great to make that connection. Miranda, I know you're sort of formally working with tree fruit and wine grape. For field bench or for berries, is soil health analysis part of what you guys are considering? Or yeah, Zach, maybe you want to pipe in, or anyone else? Also, you don't need a definitive answer, but kind of putting you guys on the spot. But it'd be just interesting to know like what the other groups are, what kind of approaches they're taking.
I don't think this type of analysis is budgeted.
Yes, that's correct.
I think having a conversation about how we coordinate in some way to have some of that analysis done would be amazing. I think there's a lot of value to me, in connecting those measurements with functional outcomes that are important to farmers. And if we could do that in multiple commodities and multiple environments, and you can see that would be amazing. But
Sean, is that Sean speaking? I can't see.
sorry, that's Sean. Yeah.
Yeah, hi. So I think that would be so powerful if we could coordinate across commodities. And it is expensive, However, it's not expensive to throw more samples on. I mean, it's not that expensive. I'm going to be doing the sequencing anyways, and we could just put more samples in with mine. So the expensive part, there would be DNA, there'd be RNA and DNA extraction. That would be, I mean, people could send me their RNA or cDNA and I could put it on with my runs. So there are opportunities where we could collaborate without a huge expense.
Yeah, and that's the question I see Hans put in the chat. How expensive is very expensive? And what is the expensive part? So like if soil samples are already happening and like where do people need to get them to before they send them to you? These are all like really important questions.
Yeah, it's all expensive. If the soils are going to be extracted for nucleic acids anyways, then that's one of the expenses it's already taken care of. The sequencing it's okay. So it's $500 a sample, which is pretty expensive, but like I said, we can't do it as intensively. So what we do is we screen the soil for areas where the communities are changing. And then we target those ones to do transcriptome analysis. So we can do more broad compositional changes, and then we just target specific communities to look at transcriptome changes. But it's expensive.
Yeah, it's expensive. But I think what I'm hearing, and I'm glad to hear it, is like we definitely need to be using this opportunity of having these different commodities as part of the living lab to make sure that for soil health, which is something that all the projects have in common, is soil, we should be doing the same analysis. So we need to coordinate that across projects. Yeah.
Yeah, I don't, in the dairy world, I don't think we necessarily contemplated
Right.
going down this path, but I guess if we did go down this path, I'm gonna put the Ministry of Agriculture staff who seem to have an envelope full of cash on the spot and say, okay, you keep saying that you've got money to spend on this. Is this important to the ministry? Then cough it up. I know, I'm a taxpayer. You paid my taxes, I paid you a whole bunch of money, so.
I didn't see the checkbox. It was like, we don't know.
So, sorry. One of them's on the screen up there. Yeah. Well, his name's up there, ain't it? Yeah. But yeah, I mean, you know what, I don't know.
Yeah, no, I love that you named that, which is great. And
Because I think at the last time the Living Lab group got together, there was a ministry staff person that said, we have bags of money.
it's true. That was part of the presentation, I think.
Yeah. it's bags of money.
Yeah.
So
go
I
ahead.
have a question for Miranda. Sorry, I don't know if
Please
anyone
do.
else is speaking.
Tag Michael Preston, we're just gonna ask a question.
Oh, yeah, no, go ahead. It's just hard to tell. Go ahead.
Yeah, absolutely.
It's just a more practical and logistical one. So one of the points of this whole workshop is how do we actually sample, right? And so sampling for soil bulk density, for soil organic carbon, you just pull the soil out of the ground. They'll be left on the ground in the baking sun. So Miranda, how are you recommending we sample for transcriptomics? Are we going to have to go out, say, with liquid nitrogen, dry ice, right?
Yes, I mean, that's what I use. I use liquid nitrogen and dry ice. You can buy RNA later solutions to put your soil in the field, which I think if we're all going to use the same thing, we should not do. We should all use liquid nitrogen. I think that's the easiest and the most appropriate. I think though that if this is something that we're really interested in, then I could coordinate perhaps my students to make some kind of tour, at least, I don't know how far, and collect the samples. Because I agree, collecting the samples, making sure they're collected in the same way is really, really important.
The furthest north is Prince George, or the.
That's not that bad. That's not too bad.
The day trip.
Delta.
No, I've done it.
That's great, thank you.
Shabtai, do you want to ask your question now?
Yeah, thanks. So I'm just wondering, we have an indication from the earlier talks and from other presentations that we've seen about these indicators of soil health, such as aggregate stability and so on, that have been mentioned, and there's other ones. So I'm just wondering if it's possible to make a connection between these microbial populations and these indicators that we know do influence the ability of the crops to grow and have resilience and so on with water stress and these kinds of things. Is there a line, a connection that we can draw? For example, I mean, what I had assumed was that fungi assist with glomalin. I mean, I don't know if I'm on a date, but with aggregates and aggregate formation, or the recalcitrant carbon. So I'm just wondering how do we make that connection between a bunch of names and numbers and complicated graphs and something that we can attach direct value to?
I mean, that's the ultimate, right? That's the holy grail, is being able to take all of these data and these. I find it overwhelming looking at these data, and I find it really hard to understand what use is. But a project like this, where we're taking all of these samples in different soil types, in different ecosystems, I think it will be a really powerful way to start addressing that, right? We'll have the mid-transcriptomic data. We'll have the actual physiological properties of the soil, the chemical properties of the soil. We'll know the composition of the soil. And so then we can start drawing some of these lines, because we really don't understand this at all. And until we do, I don't know how useful it is to continue looking at soil microbiology unless we can make that connection. And everything I do is trying to get towards that connection. And I think this is a really, really good platform to make some pretty big strides in that direction. So Shabtai, we're not there yet. Yes, fungi are important for soil stability. That's clear, for sure. Which fungi? I'm not sure. And how do our practices affect the guilds that are responsible for that? I don't know, but I think we could figure some of that stuff out here in this project.
Well, could we parse out, I know I'm going in a different direction here, but could we parse out simple questions and try to address simple questions rather than the Holy Grail all at once? Because I don't know if Living Labs is really intended for that. I mean, I might be wrong, but that's, the funding is limited, as you know. I mean, the capacity is limited, and focus is really still on carbon and greenhouse gas, right? I mean, that's still the main thing. So can we find something that we can really sink our teeth into? And I'm not in this field, so like I realize my questions may be utterly naive. So
it's not naive at all. And I think to answer that question, if what we're interested in is carbon dynamics in the soil, then we can target just the carbohydrate pathways in the soil and understand how carbon is being utilized, what guilds are active where and when and how. I think that we can absolutely focus. We don't have to look at everything. We can just pick specific areas of the transcriptome that we want to look at. But again, it depends. It depends on what ultimately, because you're right, this is a farmer-led initiative. It depends on what they really want to know about their soils.
Or as a first step, would qPCR be enough, Miranda?
Would qPCR be enough? Yeah, if we can identify, if we could identify, talk about what we're interested in. I guess qPCR is fine, but it's just a more limited picture. And we could expand it, right? We could do the entire, you could make it as big as you wanted, I suppose, but at that point, does it get to be as expensive as transcriptome? I don't know. But definitely I will follow the lead of the farmers and do what they want to do, for sure.
Well, Emma Stevens will tell us they want to make money. I'm almost sure she'll tell us that.
Okay. I don't know
Emma,
how
are you there?
Oh,
I think she's gone.
Well, maybe I can pipe in here as a producer myself. And, I don't obviously don't speak for all producers, but at least in the wine grape side, I think there's a lot of interest in some of the more longer term type of research, because we realize that there is a lot of information that we can learn from, but not today and not tomorrow, but in the longer term. And I think many of us are actually willing to invest for that longer term. So we would be, I mean, personally, I would have to say, I would be really interested in pursuing more of the transcriptomic side of research, just in order to actually develop even a baseline, and also then a data set that we can use in the future to learn from. And I think this is, I expect that this is a field of science that is going to evolve really quickly if we do put effort into it.
Okay, well, I'm gonna put as an action item that we could look at, like, what would the cost be to add this into different sites, and just sort of kind of look at the logistics of it. Yes,
Can I have one quick question?
yeah,
Again, this is from a place of ignorance, but we do have a small set of money that will be going to Luke Baynard and Lindsay Van Hoppen, who's here at the meeting today, who also use similar tools to Miranda. And Miranda, I think you and Luke know each other well. So there is a possibility of coordination, but there isn't a huge amount of money. So is there a way to take the approach that Locke suggested of being taking a qPCR approach with the money that's available, but archive these samples for the future when there is more money available, or to answer specific questions down the road?
Yeah.
Like, can we collect them ahead and then we have this bank?
I mean, I guess we could collect them and store them at -80. One of the most expensive parts of the analysis, though, is the RNA extraction and getting rid of the DNA. So I think, yeah, we could store them at -80, and we could absolutely target specific pathways with qPCR. I think that would be a really targeted, focused approach to this. And then if it answers the questions we're interested in, good enough. We don't need to do metatranscriptomics. I don't know if there's that much use in storing it. I'm of the school of thought where, the fresh with your soil, it's just everything changes over time. So I like to extract things right away, and that's the expensive step. So I don't know. I think we either have to decide, are we going metatranscriptomics or are we going a more targeted approach?
okay,
Lindsay, other question.
yes, Lindsay, go ahead.
I was just thinking, like, these are all very molecular tools that you would use, which are great, and they're definitely kind of the future of this field. But if we're thinking like logistically and limited funding wise, maybe it makes more sense to go to more basic analyses, such as just like enzyme assays, which still give you functional targeted activities, or even just like microbial biomass metrics, like carbon and nitrogen. If that, like, yeah, just logistically wise, if that makes more sense to do.
I missed the first part of what you were suggesting we do instead. I couldn't.
I was thinking more along the lines of like non-molecular tools, in line of like enzyme assays.
Yeah. No. Old school tools are really useful, really informative and useful. And I would say if you're not interested in looking at gene functioning, either through qPCR or transcripts, then I would say that would almost be more useful than looking at community composition.
Yeah.
I agree. I totally agree with that.
And Lindsay, do you just want to speak to what you guys are planning to do? You and Luke Baynard, or maybe what you.
Yeah, it's very much still under discussion. I think, once we have a bigger picture of the sites and see all the samples, our idea is to kind of pick and choose what makes sense to analyze, and whether it be qPCR and we do a simple like fungal bacterial ratio, or we look at microbial biomass, carbon and nitrogen, or enzyme assays. I think for us, anything further than that, like looking at the metagenome and metatranscriptome might be out of the picture, funding wise, but that's kind of what we're looking into right now.
I think it would be really nice if the soils across commodities could have a meeting. I would love that.
Yeah, I think that is definitely in order.